Hello everyone, Assalamu alaikum. This is me Faraz Qurban Rajpar, and in this video lecture I am going to discuss with you about the classification of penicillin. In this video lecture you will be able to learn about the different classification criteria for the penicillin, but our main focus will be on the chemical classification of the penicillin. So the classification criteria of the penicillin. There are the several criteria on the basis of which penicillin can be classified. Like one of the criteria is on the basis of the sources. Some of the penicillin are directly obtained from the natural source we have already discussed, like the natural penicillin. Some penicillin can be classified on the basis of its chemistry by the presence or absence of the certain functional group and the ring at the 6-acyl amino side chain. Some of the penicillin can be classified on the basis of its pharmacokinetic properties, like some are compatible for the oral formulation and some are compatible for the IV. Means their classification will be based on the basis of their absorption, that is the bioavailability. And some of the penicillin are the resistant to the enzyme. Means this criteria is based on the spectrum of the activity toward the enzyme, that is the beta-lactamase or the penicillinase enzyme, and on the basis of the clinical uses as well. So the penicillin, like for suppose let's consider the biosynthetic penicillin, that are the naturally occurring penicillin. The example of these penicillin are the penicillin G and B. Semi-synthetic or potentially synthetic, acidic, resistant are the stable, or what we say some are the acidic sensitive, that are can be used for the parental formulation, and some are the resistant. So these are the different criteria on the basis of which penicillin can be classified. Natural penicillin. Natural penicillin are those penicillin that are obtained directly from the natural source, that is the fungus. So the two main classes of the penicillin that falls under the natural penicillin are the penicillin G and V. They are produced biosynthetically from the Penicillium notatum or the Penicillium chrysogenum species, but mainly from the Penicillium chrysogenum species, because of the high percentage yield of penicillin being obtained from this source. So the fermentation process can be used for the production of natural penicillin. Let's consider the penicillin G. If we are going to target the penicillin G formulation or its production by the process of fermentation, then we must introduce certain chemical in our cultural media for fermentation, and that chemical compound is the phenyl acetic acid. So phenyl acetic acid is used or added to the cultural media of the Penicillium chrysogenum for the production of the benzyl penicillin, or what we say the penicillin G. Benzyl penicillin is another name used for the penicillin G due to the presence of the phenyl or benzyl ring at the 6-acyl amino side chain. There is another example of natural penicillin, that is the penicillin V. The penicillin V can be also named as the phenoxymethyl penicillin due to the presence of phenoxymethyl moiety at the 6-acyl amino side chain. If we are going to target the formulation of or the production of penicillin V, then we must introduce the phenoxyacetic acid into our cultural media of the Penicillium chrysogenum. Penicillin G. Penicillin G has a poor bioavailability. That's why it is used for the intravenous formulation. Penicillin G can be available into the various salt formulation like the sodium salt, potassium salt, benzathine and procaine. And we know that the benzathine and procaine are its organic salts, and sodium and potassium are the polar salts. Because of the penicillin G is unstable in acids, penicillin V and amoxicillin should be used for the oral formulation. So this is the reason that the penicillin G cannot be used for oral formulation. Why? Because it is unstable into the gastric acid media. So that's why it is used for the parenteral formulation. But on the other hand, the penicillin V and amoxicillin have the acid stable property, and due to this they can be used for the oral formulation. So this is the chemical structure of the penicillin G, or what we say the benzyl penicillin. You may observe that this is the beta-lactam ring and that is directly fused with this thiazolidine ring. And as we have already discussed in the SAR, that this is the position number 1. At the 2 position, the 2 methyl group are present. At the 3rd, carboxylic acid functional group. 4th position is the ring fewer nitrogen, and we have the acyl amino side chain at this 6th position, and the 7th position we have this carbonyl carbon. You may observe that in case of penicillin G, or what we say benzyl penicillin, we have the phenyl or the benzyl ring at the 6th acyl amino side chain, and due to this it is termed as the benzyl penicillin. So you may observe that this benzyl group is present at the acyl amino side chain. So due to this it is known as the penicillin G or benzyl penicillin. Why penicillin G is acid labile? We have already studied that penicillin G is acid sensitive, it is acid labile, that's why it is not used for the oral formulation. But now let's discuss that why it is acid labile and how can we make it acid stable penicillin. So there are the several reasons behind the acid instability or the acid sensitivity of the penicillins. Like one of the reason is the ring strength. The second reason is the highly reactive beta-lactam carbonyl, means the carbonyl carbon that is present at the 7th position. It's due to its high reactivity, acid instability can occur in case of the penicillin. Besides this, there is another reason, the acyl amino side chain variation, means the presence or absence of certain functional group in the acyl amino side chain. So now let's discuss these reasons one by one and their impact on the acid and stability of the penicillin. First of all, let's consider the ring strength. Ring strength is basically the type of instability. Instability means we know that molecules are formed by the bond formation. So bonds have the different stable angles, means for the stable bond formation there must be a suitable angle formed between these molecules. So once instability occur in the molecule, the reason behind the instability will be the abnormal angle, means the presence of the certain disturbed bond angle, means unstable bond angle is present. Due to that the ring strain will occur, and this is happening in the case of penicillin as well. Due to the presence of large angle torsional strain or basically the ring strain, means the instability in molecule to the presence of the ring abnormal ring, and due to this ring strain the ring opening of penicillin is going to occur, means in the presence of acidic media or what we say the gastric acid pH, the ring opening, that is the beta-lactam ring opening, will occur, and by the breaking or opening of the ring it will be converted into the unstable molecule, means its activity will be reduced, means its antibacterial activity will be retarded. So this diagram shows the ring opening mechanism of the beta-lactam ring under the acidic condition. So this is the general structure of penicillin G. You may observe that this is the side chain. Clear? So let's consider that for suppose over here the benzyl moiety is present. So once in the acidic media or in the presence of water, this will target the beta-lactam carbonyl carbon over this position, and now this will cause the change in the ring structure like this. Like over here the hydroxyl group will bond to this position, and this oxygen will ultimately transfer its electron. So what is happening, this carbonyl oxygen, this has the double bond, this bond will be transferred to this position and it has now negative charge. Clear? So once it will comes in contact with the water, then OH of water will bond to this position, And now in the next step, what will occur over here? This bond breaking will occur. Means this hydrogen will be removed, this bond breaking will occur, and ultimately this beta-lactam ring will be destroyed. Now you may observe that in the third diagram there is the ring opening, means the stable ring of beta-lactam has now completely destroyed, and due to this destruction of the ring, now its activity will be lost. So this is the ring opening mechanism that is catalyzed under the acidic condition. This carbonyl carbon becomes sensitive towards the attack of the nucleophile or the attack of the water or the acidic moiety, and now ultimately this ring opening will occur, and now beta-lactam ring has completely destroyed, and due to this its activity is retarded. So due to this is another reason for the ring opening, or behind the acid instability. So that's why it is unstable in the acidic media. So this is all happening because of the ring strain, because once it comes in contact with the acidic media, the abnormal angles are going to be formed, and due to that torsional strain, ultimately the ring opening is occurring. The second reason is the highly reactive beta-lactam carbonyl carbon. The carbonyl carbon in the beta-lactam ring that is present at the 7th position is highly sensitive towards the attack of nucleophile and does not behave like a normal tertiary amide, because the normal tertiary amide are resistant to the attack of the nucleophile, and they are going to be stabilized by the neighboring nitrogen atom. The nitrogen can fade what is happening, the nitrogen will cause or will share its lone pair electron towards the carbonyl carbon, and due to the share of its lone pair of electron towards carbonyl carbon, a dipolar resonance structure will be formed, and that dipolar resonance structure has a bond angle of 120 degree. This 120 degree is the stable bond angle that is formed in case of the tertiary amide, and due to this stable bond angle, it will become the resistance towards the attack of the nucleophile. But this resonance stabilization is impossible or will not occur in case of the beta-lactam ring, because of the increase in the torsional strain, or what we say angle strain, which will result in the double bond within 4-member beta-lactam ring. Means in case of the beta-lactam ring, the nitrogen will share its lone pair of electron towards the carbonyl carbon, but now in this case the stable bond angle, that is 120 degree bond angle, require to be resistance towards the nucleophile for the amides. But now in case of beta-lactam, this stable bond angle will not be formed, and due to this the angle torsional strain will be present, and ultimately it will destroy the activity of beta-lactam. So this is the diagram. This shows the tertiary amide. This is the chemical structure of tertiary amide. You may observe that this tertiary amide, this carbonyl carbon, and this is directly bonded to this ammonia. Now this nitrogen is going to share its lone pair of electron to carbonyl carbon, and due to this the double bond formation between this nitrogen and carbon is occurring, and it has the bond angle of 120 degree. This is the stable bond angle that is required in case of the tertiary amide. But in case of the beta-lactam, we have this amide, or what we say this carbonyl and nitrogen bond. This nitrogen will share its lone pair of electron towards this carbonyl carbon, and ultimately a double bond will be formed between this carbon and nitrogen. But now in this case the bond angle is of 90 degree, that is what we say less than in comparison to the 120 degree or the tertiary amide. So due to this decrease in angle, or due to the formation of unstable bond angle, a torsional strain will occur, and this will cause the breaking of ring, and now the structure has become flat, and due to the flatment of structure, or what we say changing of the bond angle or formation of the unstable bond angle, its activity will be destroyed. Like over here it is mentioned that the preferred bond angle for the double bond and are 120 degree. This is for the tertiary amide, clear. But the bond angle for beta-lactam ring are constrained to 90 degree. As a result, lone pair is localized onto the nitrogen atom, and the carbonyl carbon is more nucleophilic and would expect from the tertiary amide. So now what is happening? Tertiary amide, we have the stable bond angle 120, but in case of beta-lactam we have an unstable bond angle, that is the torsional strain of 90 degree, clear. And due to this bond angle, now the beta-lactam will become sensitive towards the attack of the nucleophile, and once the nucleophile will attack at this carbonyl carbon, because of its highly reactive nature, ultimately the ring opening will occur. Now let's consider the third factor that is involved in the acid instability. The third factor is the influence of acyl amino side chain. In the acyl amino side chain, we have this NH that is directly bonded with the acyl side chain carbonyl carbon. So this carbonyl carbon has a highly nucleophilic nature, and it will attack the beta-lactam carbonyl carbon like this. So this carbonyl carbon double bond will be shifted to this position, and it will have the negative charge. So in this way, this is the series of reaction mechanism will occur, and certain intermediate derivative will be formed. This is the occurring because of the destruction of the penicillin. So the different derivative like penylic acid, penicillinoic acid, and the penicillinic acid will be formed. So all these will have the ultimately effect on the antibacterial activity of penicillin is going to be decreased, and this is occurring only because of the influence of the acyl amino side chain carbonyl carbon. So now let's check out how can we make the penicillin antibiotic acid stable, or how can we make the acid resistant penicillin. Like we have already discussed the reason behind its acid sensitivity. Now let's check out how can we make them stable towards the gastric acid media. Like the acid stability of penicillin is concerned, we have discussed the three different factors like angle strain, highly reactive carbonyl carbon, and the third factor that was the acid amino side chain. We cannot control the first two factors, because if we are going to modify the carbonyl carbon, or what we say the ring strain that is occurring due to the carbonyl carbon, we will ultimately destroy the basic ring nucleus of the penicillin. So our target will be the third factor, that is the acid amino side chain. We can modify the certain functional group in the acyl amino side chain. So by the presence of the different functional group in the acyl amino side chain, we can make the penicillin stable towards the gastric acid media, and now that penicillin will become compatible or palatable for the oral formulation. Like for suppose, if we are going to introduce the electron withdrawing group in the acyl amino side chain, this will have the effect on the carbonyl oxygen, means it will cause the draw electron away from the carbonyl oxygen, and it will reduce its tendency to act as the nucleophile, because the acid instability that was occurring due to the acyl amino side chain was because of the highly reactive nucleophilic power of the carbonyl oxygen of acyl amino side chain. So once electron withdrawing group is introduced in this acyl amino side chain, now this carbonyl oxygen will become less effective as a nucleophile because of the withdrawing of electron by the electron withdrawing group from the carbonyl oxygen, and now ultimately it will not act as the nucleophile. So like in case of the phenoxymethyl penicillin, that is the penicillin V, we have the electronegative oxygen. we will observe its structure in the upcoming slide, that it will have the electron withdrawing group, and this electron withdrawing group, that is the phenoxymethyl group, will draw the electron away from electronegative oxygen, and that's why it has reduced its tendency to act as a nucleophile, and ultimately the ring opening of the penicillin will not occur, and its activity will be maintained. So in this way, penicillin V is acid stable. So if someone asks you that why penicillin V is acid stable in comparison to the penicillin G, you may answer that because of the presence of the electron withdrawing group, that is the phenoxymethyl functional group in the acyl amino side chain, and this phenoxymethyl functional group, that is electron withdrawing group, is causing the withdrawing of the electron from the highly reactive electronegative oxygen, and due to this its nucleophilic power is reduced, and due to the decrease of its nucleophilic power, the ring opening mechanism will not occur, and the activity of penicillin is maintained. The other penicillin analog that have the electron withdrawing substitution on the alpha carbon, or what we say acyl amino side chain, and are also resistant to the acid hydrolysis, like the example is the Ampicillin penicillin or the Amoxicillin penicillin. So this is the chemical structure of penicillin V, or the phenoxymethyl penicillin. So in this chemical structure, you may observe that this is the beta-lactam ring, this is the thiazolidine ring, and the all structural features are similar, but only there is a difference. This is the presence of this phenoxy ion at the 6-acyl amino side chain. So this is the phenoxy ion that will act as the electron withdrawing group. So it will cause withdraw the electron from this carbonyl oxygen, and ultimately it will decrease its nucleophilic power to act as a nucleophile and to cause the destruction of this beta-lactam ring. So in this way, the acid sensitivity of penicillin can be decreased. So that's why the penicillin V is more acid stable in comparison to the penicillin G. So this is the phenoxy. Penicillinase-sensitive enzyme. There are certain penicillin that are sensitive to what the penicillinase or the beta-lactamase enzyme. Means this enzyme will destroy these penicillin, and their activity will be ultimately destroyed. Like the example, a natural penicillin that are penicillin G and V, both are the penicillinase or beta-lactamase sensitive penicillin. They have the narrow spectrum of activity, means they will be inactive or ineffective against those bacteria which are going to produce the penicillinase or beta-lactamase enzyme. Like the example are the Staphylococcus and the Streptococcus pneumonia and the other species. Penicillinase-resistant penicillin. So this is another class of penicillin that are resistant towards the attack of the penicillinase or the beta-lactamase enzyme. Means they will not be affected by the beta-lactamase enzyme. They have the resistance power towards the beta-lactamase enzyme, and that resistance power has occurred due to the presence of certain functional group in the acyl amino side chain. Clear? So they are active against those organisms against which the natural penicillin are sensitive because of the production of the beta-lactamase. So means they will have the more power to cause the resistance towards the beta-lactamase enzyme, and now their spectrum of activity will be ultimately greater than those natural penicillin. So they are no more sensitive towards the attack of penicillinase or beta-lactamase, and they are active against all those organisms against which the natural penicillin was ineffective, and they have resistance towards the Staphylococcus species. The example are like the isoxazole derivative oxazolin penicillin that have the good oral activity, and other example are the methicillin and oxazolin. They are the example of the penicillin which have the penicillinase or the beta-lactamase resistance power. Now let's check out what are the structural features that are going to impart the penicillinase or beta-lactamase resistance power. So how we develop the resistance to the penicillinase or beta-lactamase enzyme? The reason is the presence or the introduction of the certain steric shield. Means by the introduction of certain bulky group in the acyl amino side chain, we can prevent the penicillin by the attack of the beta-lactamase enzyme. Means we have to introduce such bulky group which will bind towards the active side of penicillin binding protein and the beta-lactamase, but in this way that they can inhibit the effect of beta-lactamase and they will show the same action on the penicillin binding protein or the what we say the transpeptidase, means they are going to inhibit that. So in this way they can produce their antibacterial action. So what is the problem that can occur in the case of the steric shield? These steric shield must be not too bulky, means we must introduce the compatible functional group that can produce the favorable effect. It must not be too bulky that it can prevent the binding towards the transpeptidase as well. Means that group must be compatible towards the binding sites or binding pocket of the transpeptidase enzyme. So a great deal of work or care must be done in the selection of certain steric shield functional group. So we have the commercially available penicillin that are the beta-lactamase resistance. Now let's check out what is their structural feature and which functional group are present in their acyl amino side chain. Like methicillin is an example of penicillin that is the beta-lactamase resistance power. Methicillin was the first effective semi-synthetic penicillin that was introduced, and it has the effect against the Staph aureus species, and it has the beta-lactamase resistance power. Now let's check out what is the structural feature. It was first synthesized by the reaction of 2,6-dimethoxybenzoyl chloride with the 6-aminopenicillinic acid. Means the same 6-aminopenicillinic acid that is the basic ring nucleus of penicillin was used in the cultural media, and in this reaction media 2,6-dimethoxybenzoyl chloride was used, and in this way the newly derivative form will be the methicillin. Methicillin is also the penicillin example. So the chemical name for methicillin is 6,2,6-dimethoxybenzoamidopenicillinic acid. This is representing what? At the 6 acyl amino side chain we have this 2,6-dimethoxybenzamido derivative, and this 2,6-dimethoxybenzamido is basically a bulky functional group that is behaving like a steric shield. Means this is protecting the penicillin against the attack of the beta-lactamase enzyme. So means this penicillin will be effective against all those bacteria that are going to produce this beta-lactamase enzyme. So the steric shield are the orthomethoxy group that are present on the aromatic ring of this methicillin. So this is the chemical structure of the methicillin. You may observe that this region is the beta-lactam ring. Again, this is the thiazolidine ring. All other common structural features are same that are required for the essential antibacterial activity of the penicillin. The only difference is the presence of this 2,6-dimethoxyphenyl ring, this region, and this bulky ring structure has imparted the resistance power in the methicillin against the beta-lactamase or what we say penicillinase enzyme, because these dimethoxy functional group will act as the steric shield and they will prevent the attack of the beta-lactamase enzyme. So at the same side, if you will carefully observe that in the methicillin acyl amino side chain we have the no electron withdrawing group. So no electron withdrawing group is present, so this methicillin will be acid sensitive, and that's why it shows the poor activity against other bacterial strains. So if you will recall your memory in our previous slides, we have already studied that for making the penicillin towards gastric acid stable, it must have the electron withdrawing group as this acyl amino side chain. But you will observe there is no any electron withdrawing group present in case of methicillin. So that's why it is acid sensitive, but it is resistant towards the beta lactamase, or what we say penicillinase enzyme. The next class is the Isoxazolyl Penicillin. This is another class of penicillin, or the example of penicillin that has the beta lactamase resistant power. The incorporation of the isoxazolyl ring in the penicillin acyl amino side chain will make the penicillin again stable towards the beta lactamase. The reason will be the same. Which reason? That these isoxazole functional group will act as the steric shield. These bulky functional group will prevent the effect of the beta lactamase enzyme. So, but in comparison to the methicillin, these isoxazole have the electron withdrawing group. So beside being stable or resistant toward the beta lactamase enzyme, these isoxazole derivatives have the acid stability. The example of penicillin that comes under this class are the oxacillin, cloxacillin, phloxacillin, and the doxycycline. These are the different example of the penicillin that has the isoxazolyl ring in the acyl amino side chain. So the difference between these type of derivative, these generally are the isoxazole derivative, but their difference is the presence of the halogen substitution in the aromatic ring. This halogen substitution will ultimately affect the different pharmacokinetic parameter absorption in the body. So like in case of cloxacillin, you may observe that it has the chlorine. Clear in the aromatic ring. Phloxacillin, it has the fluorine. Dicloxacillin have the dichlorine. Clear? So now let's check out their chemical structure. This is the general ring nucleus chemical structure for the isoxazolyl penicillin. You may observe that it is the isoxazole and it is directly linked with another aromatic ring. These R substitution site are representing what the substitution of the different halogen, like cloxacillin has the chlorine, dicloxacillin has the dichloro derivative, and phloxacillin has the fluorine derivative at these sites. So the steric effect are of these bulky functional group, that is the 3-phenyl 5-methyl group of the isoxazolyl, that is the 3rd position phenyl and this 5 position methyl. These are basically what acting as the steric shield against the beta lactamase. So that will cause the prevention of binding of penicillin towards the beta lactamase active sites. Means now penicillin will be no more going to bind with the beta lactamase enzyme. Means it will have the resistance toward the beta lactamase, and in this way they will protect the beta lactam from the degradation, because the effect of the beta lactamase or penicillinase enzyme was to destroy this beta lactam ring region, and due to the destroy of this region, ultimately its antibacterial activity was destroyed. But now in this case, due to the introduction of this isoxazole ring or the steric shield, it has become stable towards the beta lactamase enzyme. So this is the isoxazole ring, and this 5-methyl and 3rd position phenyl as a steric shield. Aminopenicillin. Aminopenicillin is another class of penicillin that has expanded spectrum of activity. Means it is active against gram-positive and as well as against gram-negative bacteria, but it is inactive or ineffective against the Pseudomonas species. The examples of aminopenicillin are Ampicillin and Amoxicillin. The spectrum of aminopenicillin is due to the presence of polar functional group, that is the amino group in the acyl amino side chain, and this amino functional group has increased its penetration towards the porine cell wall channels of gram-negative bacteria, and due to this it is effective against the gram-negative bacteria as well. Now let's check out the chemical structure of the aminopenicillin. So basically these ampicillin and amoxicillin, these are the aminopenicillin, are orally active, means they can be useful for the oral formulation. Why? Because of their stability towards the gastric acid media. The stability has been increased due to the presence of polar amino functional group, and it is acting as the electron withdrawing group. As we have already discussed that whenever electron withdrawing group is present in the acyl amino side chain, then the stability of penicillin towards the gastric acid media will be increased. So both of these penicillin are acid resistant, and because of the presence of these electron withdrawing group, but at the same time they are sensitive towards the beta lactamase, or what we say penicillinase enzyme, because there is no any bulky functional group present in the acyl amino side chain of ampicillin and amoxicillin. So that's why, due to the absence of these bulky functional group, there will be no any steric shield that will prevent the binding to the beta lactamase enzyme. So that's why they are beta lactamase enzyme sensitive, and they can be destroyed by the beta lactamase enzyme. So these are the structure of the ampicillin and amoxicillin, that are the aminopenicillin. So in this chemical structure of ampicillin, you may observe that beta-lactam ring directly fused with the thiazolidine ring, and in the acyl amino side chain it has the polar amino functional group, this one. And this polar amino functional group is responsible for its extended spectrum of activity towards the gram-negative bacteria. And as well as this polar amino functional group is acting as an electron withdrawing group towards this carbonyl oxygen. So that's why it is making this penicillin as acid stable. The next structure is of the amoxicillin. Amoxicillin chemical structure is similar to the ampicillin, but the only difference is it is the hydroxy functional group para to the position of this amino. So in this structure you may observe that in the acyl amino side chain it has the amino polar functional group, which is making it stable towards the gastric acid media, and which is making it as an extended spectrum antibiotic. But at the same time it has the hydroxyl functional group, or what we say, it has the phenol ring present that is directly bonded with this amino. So these are the structure for the ampicillin and amoxicillin. So here I am sharing you the tricks to remember the complex chemical structure of the penicillin. I am quite hopeful that after this you will be able to remember, and you will be able to draw the complex chemical structure of any of the penicillin. For this you have to keep in mind some basic facts that are the common structural feature of all of the penicillin. Like, first of all, for drawing the chemical structure of any of the penicillin, first of all you have to draw the pennum ring nucleus. Pennum ring nucleus, that is basically the composition of two heterocyclic ring. One is the beta-lactam ring that is fused with the thiazolidine ring. So you have to draw this pennum ring nucleus. This is the beta-lactam ring, and this one is the thiazolidine ring. In the next step you have to keep the dimethyl group at the position number 2, like this. Over here at the position number 2 the dimethyl group is present. The third point is you have to place the carboxylic acid group at the third position, and then you have to keep the acyl amino side chain at the 6th position, like this one. This is the amide linkage, that is the carbonyl group is directly bonded with the amino group. Clear? So these are the common structural features of any of the penicillin. You have to keep in mind this entire basic ring skeleton for drawing the chemical structure of any of the penicillin. Clear? So every penicillin is differing from one another by the substitution of this group. Only this R is different in each of the penicillin. Like in case of penicillin G, we have the benzyl group at this position. In case of penicillin V, we have the phenoxy group at this position. So first of all, you have to draw this entire ring basic skeleton for drawing the chemical structure of any of the penicillin, and you have to substitute this R according to the your desired penicillin. So if you are going to draw the chemical structure of penicillin G, you have to replace this R with this benzyl group. Clear? If you are going to draw the penicillin V, then you have to draw this phenoxy moiety at this position, and so on. In case of ampicillin, you have to place this benzyl moiety and then again place the amino group at this position, and same in case of amoxicillin and methicillin. So you have to keep in mind this entire basic ring skeleton for each of the penicillin, and you have to substitute this R with these groups. Antipseudomonal penicillin. Antipseudomonal penicillin are those penicillin that have the activity against the Pseudomonas species. Now let's check out why they are active against the Pseudomonas species. Because of the presence of the carboxylic acid functional group in their acyl amino side chain. That's why the antipseudomonal penicillin can be commonly termed as carboxy penicillin. The example of antipseudomonal penicillin are carbenicillin, tetracycline. So carbenicillin was the first example of this class that was introduced, or what we say by synthesized. It shows a broad spectrum of activity towards the pseudomonal species because of the presence of hydrophilic carboxylic acid functional group in the side chain. So it is the broader range of antibacterial activity than any of the other penicillin and that property is due to the presence of this carboxyl functional group. This carboxyl functional group has imparted the increase of the penetration power through the cell wall barrier of the gram-negative bacteria than other penicillin. So now they can be easily penetrate into the gram-negative bacteria bacilli, and due to this their spectrum of activity has been enhanced. But the carbenicillin are not stable in the acids, means they are inactivated by the acidic media, and beside this they are also destroyed by the beta-lactamase, means they do not have the beta-lactamase resistance power. So this is the chemical structure of carbenicillin. You may observe that in the acyl amino side chain we have this carboxyl functional group present, and there is no any electron withdrawing group present near to this carbonyl carbon, so that's why it is acid sensitive. And besides this, there is no any bulky functional group that will going to be act as what we say steric shield, and that bulky functional group is absent in case of carbenicillin. That's why it is sensitive towards the penicillinase or beta-lactamase. So you may observe that all of the properties of carbenicillin is due to the presence of this carboxylic acid functional. Uridopenicillin are the class of penicillin that have the polar functional group in the acyl amino side chain. The examples are the piperacillin and the azoxyllin. Piperacillin has the piperazine ring in the acyl amino side chain, and the azoxyllin has the imidazole ring, but they have further different substitution or the functional group or the other ring attached to these rings, means piperacillin has the piperazine ring and the further substitution on that piperazine ring. They have the greater or the extended spectrum of activity, like they have the activity against the Pseudomonas species and as well against the Klebsiella species, and they have the good potency against the gram-positive bacteria, but generally they are ineffective or what we say sensitive towards the beta-lactamase enzyme. So this was all about the classification of the penicillin.